Hello, everyone. I'm Philip Yusuf. I'm going to be working with Dr. Wolf. We worked on establishing novel salivary flow cell capabilities to study the oral microbiome. Here's my introductory slide here. The dental health is a good indicator of our overall health, and there are several oral pathogens that can affect the dental health. In the oral microbiome, there are over 700 species of microorganisms that live and exist together, creating a community and they can form biofilms to increase their survivability as well as their viability. So in the image on the right here, in the image right here, we can see just some of the common examples of bacteria you find in the oral cavity. You can see the strep mutants here, and you can see the interactions of how it inhibits as well as facilitating different other kinds of bacteria. And then we can see some other examples here as well. In this study, we hope on providing some valuable insights into how the variability of bacterial species can be used in clinical diagnostic as well as preventative care. We plan on using conventional and novel techniques to study biofilm formation as well as variability in bacterial species. So here, just talking a little bit about our methods and how we went about with our data collection. So we had a total of 5 volunteers who also work in the same laboratory. We collected some saliva samples using a passive drill system, using salivettes, and we also tried to get bacteria using a floss method. And then we purified and isolated DNA using the cryogen DNA-seq kit. And then we subjected our samples to PCR amplification for the 16S ribosomal DNA. And this gene is present in all bacteria. So here we can see the white parts, which is constant to every bacteria. And then we can see the variable regions from V1 through V9, which is different depending on the 16S region of a particular DNA. And so we use universal primers 8F and 1492R to amplify this particular region. So moving on to my next slide, we got the DNA concentration of our floss DNA samples as well as our saliva samples. So here we can see the floss, we had less DNA concentration compared to the saliva samples. We predicted that the saliva might have had more total DNA due to the fact that there was more mammalian cells present. We also checked the purity of our sample as well. So here, we expect purity to be within the range of 1.7 and 2.0. So most of our sample fall in that range with the exception of one. So the absorbance is at 260/280 for pure DNA. And so moving on to our next slide is our gel electrophoresis of our PCR product. So here we can see our 3 different saliva samples. We can see we have the expected 1,500 base pair product of that 16S DNA. And then in our floss samples, we can see we only saw that band in one of those lanes. And we predicted that this might be due to the fact that we had low DNA concentration for the floss samples to begin with. And this was done in a 3% agarose gel. And then next to it, we performed a DNA digest. We used the BST restriction enzyme, which is a 6 base pair cutter. So here's our 3 samples showing the various restriction fragment length polymorphisms. And each of those bands are different depending on which 16S DNA bacteria you're looking at. So here we have the floss samples again. We don't see much there as well, due to the fact that we had low DNA concentration there. And so moving on to my next slide, we reran the experiment, but this time around we used just the saliva samples. So in the first 4 sample lanes, we can see the genomic DNA samples, which is what we expected, right? We expected a 23,000 base pair product there. And then in the next 4 sets of samples, we can see our observed and desired 1,500 base pair PCR product for the 16S ribosomal DNA in the 1% agarose gel. And then next to it, we use a different restriction enzyme, which is a 4 base pair cutter. That is the HA3. So we can see the 4 different samples, right? So we have the ladder, and then we have our DNA digest 1, 2, 3, 4. These are all 4 different human samples. And we can see some variability from person to person, suggesting the presence of different bacterial species. So these are all RFLPs, showing us the diversity of our microbiome. So this next slide talks about the microincubator we plan on implementing for the study. So my PI, Dr. Wolf, he designed this from scratch, and he made this for the hopes of being able to regulate conditions such as carbon dioxide, oxygen, temperature and the like. So here we see the nitrogen tank as well as the carbon dioxide tank here. We want to be able to regulate those conditions to areas that are also found in the oral cavity. And so this microincubator also houses a salivary flow cell chamber, which is shown here. So the chamber has, this is the chamber, and then this is a syringe pump. In the chamber, we can see the inlets. And for this experiment that we did, we seeded the inlet with bacteria from fresh saliva. And then we loaded the chamber with a low ACU gel and gum at 0.5%. And then here we have the syringe pump in which we filled with a sample of 1:4 ratio of pure saliva and Krebs Henselite buffer, which is a physiological buffer. The pure saliva was centrifuged and the supernatant was filtered with a 0.2 micrometer filter. And then the saliva here serves as the sole nutritional source. I run it at a very slow rate of about 0.4 mil per hour. And so that's what we did. And so in the long term, we do plan on following through with this hypothesis to where we said that biofilms formed close to fresh saliva in the flow cell chamber would be different from those from further downstream where survival of the fittest would have occurred, as preferred nutrients in saliva would have been diminished. And so here we took out the cover slip and we observed that under an upright microscope, which is shown in this next slide here. So this is just an example of a biofilm we're able to see. Here we see the different shapes of bacteria suggesting the presence of different bacteria within the biofilm. We can see some rods here, some chains, some calculi, and some chains here again. And then we tried isolating the DNA from the gel, but we were unsuccessful in doing that. We plan on optimizing our techniques so we can better visualize our desired PCR product here. And so that brings me to some of the limitations we had. I already mentioned this point about optimizing the DNA isolation protocol. We also plan on using sunset yellow, which is a vital stain, as opposed to the gram stain we used for the previous image I showed. And we plan on using this in a clear chamber so we can better visualize the biofilm formation. We also plan on using a peristaltic pump as opposed to the one we use in this study. The peristaltic pump is going to help us run the saliva and the KHP buffer at a slower rate for days as opposed to just 12 to 24 hours that we run this for, so we can better visualize our biofilms. And here are just some conclusions. We're able to see some variability in the bacterial species across various human samples. And we also developed the saliva flow cell chamber to visualize biofilm as well. And in the future, we plan on creating an isolated local environment with varying levels of temperature, carbon dioxide, oxygen, as well as different sugar concentrations like sugar, glucose, and xylitol. And we and that future studies would implement techniques like NGS to observe the variabilities in the oral microbiome. And so here are just some of my references and then my acknowledgements to the special text necessary for the funds for this research. It's like my mentor, Dr. Wolf, or my partner, Moliker, will also help a lot in this research, and then Dr. Stodinger, and Dan. Yeah, thank you.